This is chapter 23. Here we're going to be looking at the evolution of populations. So this is going to look at the smallest units of evolution. A common misconception is that organisms evolve during their lifetime. They don't. Natural selection acts on individuals, but only populations can evolve. For example, a population of medium ground finches on Daphne Major Island. During a drought, large-beaked birds were more likely to crack large seeds and survive. The finch population evolved by natural selection. So what you ended up with is looking at the birds before and after the drought. 3 years earlier, the size of the beak versus after the drought, the size of the beak was larger and changed. So it wasn't that the individual birds had their beaks grow larger. It was that those with better beaks, larger beaks, were more likely to be successful and propagate and pass those genes on to the next generation, so that you ended up with larger beaks being in future generations. So here between 1976 and 1978 is a relatively short time for something to evolve, but birds also have fairly short lifespans. So microevolution is a change in allele frequency in a population over generations. There's 3 mechanisms that will cause the allele frequency to change: natural selection, genetic drift, and gene flow. Only natural selection will cause adaptive evolution. Variation in heritable traits is a prerequisite for evolution. Mendel's work using the pea plants provided evidence of the genes being the discrete heritable units. So genetic variation among individuals is caused by differences in the genes or other DNA segments. Your phenotype is the product of the inherited genotype and the environmental influences. It's going to be what the organism looks like. Natural selection can only act on variations with a genetic component. So if it doesn't have a genetic component that can be passed on, natural selection won't be involved. Some phenotypic differences are determined by a single gene, and they can be classified on an either-or basis. Other phenotypic differences are determined by the influence of 2 or more genes, and they vary along a continuum within the population. So here you can see in these horses, there isn't a dark-colored or light-colored horse. There is a continuum of colors of those horses. So here you can see that you have recombination that's going to occur to actually create your genetic variation. So here, when you have recombination, you're going to get different combinations of alleles that will be passed on to the offspring in the future. So you can have base pair substitutions, and you can have these sites where you have insertions. So the substitution results in translation of a different amino acid. Some phenotypic variation does not result from genetic differences among the individuals, but rather environmental influences. Only genetically determined variation can have evolutionary consequences. So sources of new genetic variation. You can have new genes and alleles arise by mutation or by gene duplication. Sexual reproduction can result in genetic variation. It can recombine existing alleles. So you can get portions of an allele from one parent and portions from another. A mutation is a random change in the nucleotide sequence of the DNA. So only mutations in cells that produce gametes get passed on to offspring. So for example, if you look and you get a mutation in some of the skin cells that are on your hand, those are not going to be passed on to your offspring. It would only be passed on to the offspring if it occurs in the sperm or the egg. So a point mutation is just a change in one base in a gene. And the effects of the point mutations can vary. Mutations that result in a change in a protein production are usually harmful. It's usually not going to make a protein that works as well as the original. The harmful mutations can be hidden from selection in recessive alleles. So if it's in a recessive allele, it won't actually show up in the phenotype. The individual would not even be aware that it's there. Mutations that result in a change in protein production can sometimes be beneficial as well. So point mutations in non-coding regions generally result in neutral variations. It's not going to give any selective advantage or disadvantage. It's going to work just as well. Mutations to genes can be neutral because of the redundancy in the genetic code. So if you have a mutation and both of the mutated and the non-mutated gene are going to start coding for the same amino acid, it's going to be a neutral mutation. Chromosomal mutations that delete, disrupt, or rearrange many loci are typically harmful. Duplication of small pieces of DNA increases the genome size, and that's usually less harmful. Duplicated genes can take on new functions by further mutation. So, an ancestral odor detecting gene has been duplicated many times. For example, in humans, we have 350 copies of the gene. Mice have 1,000 copies of the gene. So mutation rates are relatively low in animals and plants. The average is about 1 mutation in every 100,000 genes per generation. The mutation rates are often lower in prokaryotes and higher in viruses. Mutations will accumulate quickly in prokaryotes and viruses because they have short generation times. Sometimes their generation time may only be 20 minutes. Sexual reproduction can shuffle existing alleles into new combinations. So in organisms that reproduce sexually, recombination of the alleles is more important than mutation in producing genetic differences that make up adaptation possible. So you're going to just get more variation due to sexual reproduction because they don't have that many mutations. The first step in testing whether evolution is occurring in a population is to clarify what we mean by a population. So a population is a localized group of individuals that are capable of interbreeding and producing fertile offspring. The gene pool is going to be all of the alleles for a loci in a population. If the locus is fixed, all the individuals are homozygous for the same allele. They're all going to have the same allele. So here we can see where you've got a porcupine herd range and a 40-mile herd range, and this area where they overlap. The Hardy-Weinberg equation is a way of describing the genetic makeup we expect for a population that is not evolving at a particular locus. So if the observed genetic makeup of the population differs from the expectations under the Hardy-Weinberg, it suggests that the population may be evolving. So in a population where the gametes contribute to the next generation randomly and Mendelian inheritance occurs, the allele and genotype frequencies remain constant from generation to generation. So we say that that population is in Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is a population that's not evolving. So the Hardy-Weinberg theorem describes a hypothetical population. Real populations, the allele and genotype frequencies do change over time. So there are 5 conditions to have a non-evolving population, and these are rarely met in nature. First is that they have no mutations. Second is that they'll have random mating. Third is no natural selection. 4, an extremely large population size. And 5 is no gene flow. So natural populations can evolve at some loci while being at Hardy-Weinberg equilibrium at other loci. So there can be some traits that don't appear to be evolving while others are. There are 3 major factors that alter the allele frequency and bring about the most evolutionary change: natural selection, genetic drift, and gene flow. The differential success in reproduction results in certain alleles being passed to the next generation in greater proportions. So for example, an allele that confers resistance to DDT in fruit flies is going to be seen in increased frequency after DDT was widely used in agriculture. Those are the ones that survived that went on to breed. Natural selection can cause adaptive evolution. It's an improvement in the match between the organism and their environment. They're better suited. So the smaller a sample, the greater chance of random deviation from a predicted result. Genetic drift is going to describe how allele frequencies fluctuate unpredictably from one generation to the next. Genetic drift tends to reduce genetic variation through the losses of alleles. So if we look here, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 plants. 5 plants are going to leave offspring. So when we look at the variation in the offspring, you went from having 5 that are highlighted in yellow here to only 2. These 2 plants are going to leave offspring. When you look at the color proportions, the frequency of having red or white or pink flowers, you go from having a mix to having more red, and just depending, if you had 2 reds that were heterozygous for being red, the next generation you end up with all reds. With the founder effect, this occurs when a few individuals become isolated from a larger population. So if you were to send a few individuals to a new land to survive, the allele frequencies in the small founder population can be different from those in the larger parent population. A bottleneck effect is a sudden reduction in the population size due to a change in the environment. So your resulting gene pool may no longer be reflective of the original gene population. So if the population remains small, it may be further affected by genetic drift. So here you can see your original population. There are some actual little yellow ones in that population. With the bottleneck effect, you don't get an equal sample of all of them. Not everybody that was in the original population is going to be represented. So your surviving population here doesn't include any yellow ones. So understanding the bottleneck effect can increase the understanding of how human activity affects other species. So loss of prairie habitat caused a severe reduction in the population of the greater prairie chickens in Illinois. The surviving birds had low levels of genetic variation, and only 50% of their eggs hatched. So here you can see this greater prairie chicken, the range of the greater prairie chicken pre-bottleneck, and then the range post-bottleneck where it's much smaller. So then you can look at the number of alleles per locus and compare them, and the percentage of the eggs hatched and compare them. So they go way down here. So the effects of genetic drift, genetic drift is going to be more significant in small populations. It can cause the allele frequencies to change at random. It can lead to a loss of genetic variation within the populations, and it can cause these harmful alleles to become fixed. Gene flow consists of the movement of alleles among populations. Alleles can be transferred through the movement of fertile individuals or gametes, such as pollen. Gene flow tends to reduce variation among populations over time. Gene flow is an important agent of the evolutionary change in modern human populations. So natural selection is the only mechanism that will consistently cause adaptive evolution. So evolution by natural selection involves both chance and sorting. So new genetic variations arise by chance, but beneficial alleles are sorted and favored by natural selection. So natural selection consistently increases the frequencies of the alleles that will provide a reproductive advantage. So relative fitness, the phrase struggle for existence and survival of the fittest are a little bit misleading because they imply direct competition among individuals. Reproductive success is generally more subtle and it depends on many factors. So relative fitness is the contribution of an individual that they make to the gene pool of the next generation. So the relative contributions of other individuals. Selection favors certain genotypes by acting on the phenotypes of the individuals. So there's 3 modes of selection. Directional selection favors individuals at one extreme end of the phenotype range, so just one extreme or the other. Disruptive selection favors the individuals at both extremes of the phenotypic range. And then stabilizing selection favors the intermediate variants, and it acts against the extreme phenotypes. So for example, if we have here a range of fur colors in the original population, With directional selection, you're going to have the dark ones, for example, be favored in the evolved population. With disruptive selection, it's the really light ones and the really dark ones that are favored. And with stabilizing selection, it's the medium colored ones that are favored. So striking adaptations have arisen by natural selection. For example, certain octopuses can rapidly change color for camouflage. The jaws of snakes allows them to swallow prey larger than their heads. So here you can see the bones that are movable are actually shown in green. So this egg is larger than the snake's head. You can open it up and swallow it. So natural selection increases the frequencies of alleles that enhance survival and reproduction. Adaptive evolution occurs when that match between the species and its environment increases. They're better adapted to their environment. Because the environment can change, adaptive evolution is a continuous process. Genetic drift and gene flow do not consistently lead to adaptive evolution, as they can increase or decrease the match between an organism and its environment. Sexual selection is natural selection for mating success. It can result in sexual dimorphism, where you have marked differences between the sexes in their secondary sex characteristics. So here you've got two peacocks, and there's a marked difference between the appearance of the male and the female bird. Intrasexual selection is direct competition among individuals of one sex. This is usually the males that will have to compete for the mates of the opposite sex. Intersexual selection, this is often called mate choice, occurs when individuals of one sex, usually the females, are choosy in selecting their mates. So male showiness due to mate choice can increase a male's chances of attracting a female while decreasing his chances of survival. So how do female preferences evolve? The good genes hypothesis suggests that if a trait is related to male genetic quality or health, both the male trait and the female preference for the trait should increase in frequency. So diploidy maintains genetic variation in the form of recessive alleles that are hidden from selection in the heterozygotes. With balancing selection, this occurs when natural selection maintains stable frequencies or two or more phenotypic forms in a population. So balancing selection includes heterozygote advantage and frequency-dependent selection. So the heterozygote advantage occurs when heterozygotes have a higher fitness than do both homozygotes. So natural selection will tend to maintain two or more alleles at that locus. And the heterozygote advantage can result from stabilizing or directional selection. So one case of this that is a common example is a mutation in the allele that codes for part of the hemoglobin protein that causes sickle cell disease. But this will also confer malaria resistance. So in the regions where the malaria parasite is common, selection favors individuals who are heterozygous for the sickle cell allele. They are more likely to survive. So when you look events at the molecular level, the sickle cell allele on the chromosome here, an adenine replaces a thymine, and you end up with a sickle cell hemoglobin. So under low oxygen conditions, it will become a sickled red blood cell that is not going to fit through the blood vessels very well, and that's what's going to cause symptoms and disease in the individual. Here, when you have the normal wild type allele, you have normal hemoglobin, normal shaped red blood cells that are going to fit through. So these are also easier for malaria to attack. These are more resistant to malaria. So when you look at the sickle cell allele and evolution in populations, when you look at the distribution of malaria and then you look at the frequencies of sickle cell allele, they overlap. So this is the Anopheles mosquito. This is going to be the one that transmits malaria to people when they bite them. In frequency-dependent selection, the fitness of a phenotype declines if it becomes too common in the population. So selection favors whichever phenotype is less common. So for example, the frequency-dependent selection results in approximately equal numbers of right-mouthed and left-mouthed scale-eating fish. So you have these right-mouthed and left-mouthed fish. If you have too many right-mouthed fish, there is not going to be enough of the left side of this fish available for all of them to eat. So if you are a rare fish that can eat off the other side, you're going to have more food available and less competition. So natural selection cannot fashion perfect organisms. It can only act on existing variations. Evolution is limited by historical constraints. The adaptations are often compromises. They're not necessarily ideal. So you have chance, natural selection, and the environment all interacting together.